right. Well, thank you, and I'd like to thank the organizers for the invitation to speak today, and thank all of you for sticking it out until the end of the program today. We just need to hook up my computer there. There it is. Okay, great. Okay, so let's see. Is that? Yeah, all right. There we go. Okay. Oh, now it's responding. Sorry. Let me go back to the beginning. Or could you put it back to the beginning for me? Thank you. There's a delay. There we go. Okay, so Regenerative Patch Technologies, LLC, is a spin-off of a CIRM-funded disease team project, which hopefully is going to come up in the second slide. There we go. The company was formed in 2011, and it's based on what we call the California Project to Cure Blindness, which is a collaboration between USC, UCSB, Caltech, City of Hope, and University College London as a partner PI. It's led by Mark Humayun, who's at the USC Eye Institute, and myself and David Hinton are co-PIs on the project. The focus is to advance stem cell-based therapies for the dry form of AMD and carry forward the work started in the California project. We were awarded a CIRM Disease Team 1 in 2010, which is just finishing up, and we're about to start a CIRM Disease Team 3 grant to fund the Phase 1/2A clinical trial. Let me tell you just a little bit about the disease, we're focused on the dry form of AMD. If you look at the picture on the left, this is a normal fundus photo. When you go into the ophthalmologist or optometrist and get that bright flash, they're taking a picture of the surface of your retina, and that's what it looks like. In the disease, it can progress to an early form of AMD characterized by these little yellow dots that you see, which are called drusen, and then it can go either to the dry form or the wet form and in advanced disease. Now, there are good treatments for the wet form, which involves improper growth of blood vessels, and you can block that with antibodies or inhibitors of VEGF. But for the dry form, which is 80 to 90% of the cases, there's really no good option for patients. Now, we know a little bit about the disease, and most types of the disease are characterized by dysfunction or death of the RPE, retinal pigmented epithelium, and degeneration of the Brooks membrane where the RPE sits in the eye. The RPE cells are really important for supporting photoreceptors, and as a result of the RPE loss, you lose photoreceptors, and that leads to vision loss. So our treatment hypothesis is that we can replace the damaged RPE and prevent that loss of vision. Now, just a little bit more about the RPE. You can actually see the RPE when you look into somebody's eye and you're looking at the pupil. It's black. You're actually looking at this pigmented monolayer of cells behind the retina, as you can see in this cartoon here. And they're a polarized monolayer that extend apical microvilli that interdigitate between the rods and cones. And they're super important for vision. Let's see if I can go one more there. First of all, they absorb stray light, provide a black background. And every morning when light hits your eye, they carry out phagocytosis of shed outer segments of the rods and cones. Super important activity that if you don't do that, you'll go blind. The RPE also transports nutrients from the choriocapillaris, and if you look at the blood supply to the eye, shown here on the right, the choriocapillaris is just on the basal side of the RPE, and if you look at where the outer segments of the rods and cones are, there isn't a lot of vasculature. So RPE has a really important role of moving things from the blood into that area of the outer segments. Oops. If we go back 1. There we go. The other really important thing is that every time light hits your rhodopsin, you use a molecule of retinol, and it's isomerized, and the retinoid cycle recycles that, and the RP is involved in that. So really crucial cell for function of the RPE. Now, macular degeneration is very common. Something like 3 out of 10 people over the age of 75 have some form of the disease, and it's estimated that 7 million or more people in the U.S. Suffer from this disease. Now, it's an opportunity, and as I said, 80 to 90% of the cases are the dry form. If you look at Lucentis sales, and if we predict or project a price point similar to a year's worth of Lucentis treatment, we can see quite a large U.S. market potential. And guess what? A lot of people think this is a good idea, and there are a number of groups going forward with therapies for dry AMD. You've probably heard about the ACT trial that's ongoing. Earlier today we heard about Stem Cells Inc., and you probably saw in the news in Japan they just implanted 3 patients with iPS-derived RP. So there are a number of strategies using a number of different cell types, some in suspension and some on implanted as monolayers. And our approach is a little bit different than others. What we're doing is to take human embryonic stem cells, make RPE, then grow them on an ultra-thin membrane and as a polarized sheet of cells that's then implanted in the eye. And I'm just going to mention a few advantages of this approach. We know that for RP function, the cells have to be polarized, and studies have shown that polarized cells behave a lot better than suspension cells. For example, if you look at the secretion of the factor PET-F, which is a support factor for photoreceptors on the left, polarized RP make tons of PET-F and secrete it from the apical side. But David Hinton's group has shown that non-polarized RP do not secrete PET-F. We also know that in the disease there's a breakdown of Bruch's membrane where the cells normally sit. And so our approach is to provide them with a synthetic Bruch's membrane. It's a parylene membrane. Parylene is a USP class 6 biocompatible polymer, xylene polymer. And it's been engineered by our collaborators at Caltech to mimic the normal Bruch's membrane in terms of permeability, such that nutrients can get across from the choriocapillaris to the RPE. And we coat that with vitronectin, which we think provides an advantage to the cells after implantation. And recent studies have shown from our group that if you compare a bolus injection of HESC RP to cells on a substrate, the cells on the substrate survive longer in the nude rat model. So we think we're basically providing them with a home so that they'll survive better and also function better. They're going to be implanted such that they're in the right orientation as a polarized sheet. Now we've come along in our approach to this and have now completed cGMP production of enough cells for the phase 1 clinical trial with our partners at City of Hope. And this slide just shows our process where we grow the cells, differentiate them, enrich them, expand them, and then cryopreserve as an intermediate cell bank. And this gives us a real advantage because we have a stopping point. Then the final product is prepared by thawing out the ICB and growing it for 30 days on the parylene membrane, which is then shipped to the point of care. We've developed this parylene membrane as a 3 by 5 millimeter piece of parylene with these ultra-thin regions so that diffusion is normal compared to a normal Bruch's membrane. And we estimate about 100,000 cells are required to cover each implant. So the differentiation process takes about 6 months, and it's actually quite simple in that we just remove the FGF from the human embryonic stem cells. And the cells start to differentiate into a number of different cell types. But if you look at panel A in the picture, with time, you can start to see pigmented foci, and we've worked to identify specific substrates and media formulations that give rise to high levels of differentiation. And we can pick these manually and also use an enzymatic treatment to enrich the cells, which are shown in panel B after enrichment. And using a selective media, we can grow these up such that the RPE have a growth advantage over contaminating cells, and you end up with very nice homogeneous RPE shown in the bottom right. And if you stain for the RPE marker PMEL shown on the right, you can see that 99% of the cells are positive for PMEL markers, or for RPE markers. So, and very importantly, if we look for potential contaminating cells, we don't see any undifferentiated HESC markers left in the population. So it's a stable, fully differentiated and polarized cell sheet. They're not dividing. They're held together by tight junctions, so we don't think they'll go anywhere. And they're functional. We can show that they carry out phagocytosis in vitro and secrete PEDF, similar to the levels we see in fetal human RPE. So proof of concept is the same model that Henry just described, the RCS rat model. And this model, this rat has a mutation in a receptor important for that phagocytosis that I told you about. And while the rats are born with photoreceptors, they degenerate after 12 weeks. And so we were faced with a challenge here because our implant is not an injection of cells, it's cells on a scaffold. And the rat eye is pretty small. So we couldn't go in as early as what Henry showed. We implanted about postnatal day 28 and then look over time to measure whether or not the cells are phagocytosing outer segments, to look at photoreceptor survival and visual function over time. And I'll just show you some of the data. One thing we were interested in is, do the cells survive? And if you compare cells on a scaffold versus a bolus injection, you can see on the left-hand side good survival up to 6 months in the RCS rat, whereas the bolus injection, we could see what looked like injected cells, the pigment shown by the white arrow, but those turned out not to be positive for human antigen, and in fact, we think are macrophages that have eaten the dead cells that did not survive well in the RCS rat. So we can quantify the phagocytosis by looking at rhodopsin staining, and shown in the left are phagosomes in the implanted RPE carrying out phagocytosis. And as Henry mentioned, we can do recording in the superior colliculus and show, if I'll just skip to the next one, that the parylene with cells has a better response than parylene alone or the control. So how do we work out the surgical method? Well, the rat eye is a very different surgery. So we go to the pig eye, which is about the same size as a human eye, and this is a movie made by Rodrigo Brandt and Mark Humayun of a surgical procedure to implant the device in the Yucatan mini pig. And what you're looking at is a movie made by looking through the lens down into the eye. They remove the vitreous and then inject some fluid to create a bleb that separates the retina from the RPE. And here's our scaffold covered with cells. We've developed a special tool where the scaffold can be pulled inside a cannula and it folds up kind of like a taco shell, and then that's inserted through the side of the eye and then through a small retinotomy in the bleb, and the implant is then extruded and it unfolds behind the retina in the area where the RPE cells in a patient would have degenerated. So we think we can do this as an outpatient procedure in about 45 minutes, a 1x treatment that will ultimately, we hope, help patients. Let's see if, there we go. Let me just skip to the next slide. So we're on our way to filing an IND. We're in the final preparation of the IND right now. We had our pre-IND meeting back in 2012. We've carried out preclinical proof of concept studies, the IND-enabling safety and toxicity, and as I said, we've completed the GMP production of cells. So we're planning, hopefully next year, to start a phase 1 trial looking at patients with dry AMD, and it'll be a safety trial as the primary endpoint, and secondary to look at visual function. So that's where we are now, and I'll finish up there and just like to thank my collaborators at all the institutions involved, the California Project to Cure Blindness, and especially funding from the California Institute for Regenerative Medicine. Thank you. Questions? Any questions? Yeah. Yes?
How long does the patient need to have systemic immunosuppression?
Say that again?
How long the patient needs to take systemic
Yeah. We're planning to use systemic immunosuppression following the lead of ACT. We don't know that we'll need it, and in fact, in some of the preclinical animal studies, we have not used immunosuppression and have not seen rejection of the cells. You know, in theory, it's an immunoprotected site in the subretinal space. But in patients, we really don't know what to expect. And some patients may have a compromise in their blood-brain barrier, and we may need the immunosuppression. So that's something that really we need to explore further. Yes, here. And then.
When you transplant the cell with this padding membrane, is that where so the cell won't expand after you transplant?
That's right. That's right. These cells are not dividing. They've formed this monolayer, have established their tight junctions and have polarized. And in fact, in vivo, the normal RP does not turn over very much at all. So we like that. We want to deliver them to where they're needed, and we don't want them to wander around and go cause trouble somewhere else.
So when in your clinical trial, when you try to transplant these pieces, are you going to transplant multiple pieces for some patient maybe have a large area?
Yeah, this piece is big enough to cover the macular region, and so we're hoping for a one-size-fits-all type of approach and a one-off treatment. Yeah, Henry.
You mentioned that your colleagues had looked at the scaffold and constructed it in a way that took into account permeability issues. I thought that was really important. I just thought maybe you could elaborate a little on how it was done.
Yeah, this work was carried out in Y.C. Tai's lab at Caltech, who is a bit of a parylene expert. And using nanolithography methods, they were able to develop and test different formulations and look at permeability in vitro, just looking at the diffusion of different size molecules across the parylene. And what they came up with was a mesh that's supported by 6-micron regions with ultra-thin regions that are 0.5 microns. And the challenge was to make it thin enough, but also strong enough to survive the surgical manipulation. And there was a lot of back and forth between the engineers at Caltech and the surgeons, and this is what they came up with. So it's robust and survives surgery. We've done experiments in human cadaver eyes. The FDA wanted us to put it in and take it out, load and unload, and look and see if there was any stress on the cells, look and see if there was any damage to the membrane or loss of cells, and we see good retention of cells and no damage to the membrane.
Are you thinking of this as a permanent implant?
Yes, that's a good point. I forgot to mention this is a biostable substrate, so it won't biodegrade, and we imagine it staying there. Yes, and at first, you may think, "Ooh, I don't know if I want a piece of plastic stuck in my eye." But if you think about the most common eye surgery for cataracts, you get a plastic lens, and it stays there and does just fine. So. Yeah, would it interfere with the choriocapillaris? And that's something that's very important, and there is loss of choriocapillaris in AMD and geographic atrophy. we hope that we won't see any loss of the choriocapillaris. Our cells secrete VEGF from the basal side, so we think they'll be constantly delivering that to keep the choriocapillaris alive. And you know, in some cases there may even be a double layer where we've put this over some existing RPE. But what we imagine is sort of a two-part transport system where the endogenous RPE would transport things, and then it would be picked up by the implant and transported across. So we're hoping that that will work well, and as I said, we do see good survival and rescue in the animal models. Okay, thank you very much.